My name is Luca Vangelista. I am a group leader at the San Raffaele Scientific Institute, and I'm part of the CHAARM consortium. And I work on derivatives of CCL5 rantes, that is a chemokine, a natural protein that every one of us has in the body, which has been discovered as an inhibitor, a natural inhibitor of the entry of HIV virus into the target cells. And because of this, it has been an important compound to study in order to develop derivatives which can be improved for their anti-HIV activity. And my approach to this field is to develop two different categories of derivatives that are short peptides of the protein and full-length mutants. For the short peptides, I am fully involved in the CHAARM consortium, and for the full-length mutants, I am involved in different contexts, but all of them are related to microbicides. In regard to the peptides, I have continued a long-lasting project that started more than 10 years ago, in which we have developed a molecular evolution of these peptides by modifying some key residues. And at the moment we have the best compound that has an anti-HIV activity that is comparable to T20, that is actually a drug used in therapy, which blocks the entry of the virus by freezing conformation of the proteins of the virus that actually interact with the cell target. So this is very important because it's a proof of principle that you could have new peptide-based drugs. And within CHAARM, I'm collaborating with several other partners, particularly with Stefan Grzesiek on the characterization of the interaction of rantes derivatives with CCL5. And I also collaborated with Dominique Scholz, who has tested a Maraviroc-resistant strain of HIV, where Maraviroc is actually a drug that binds to CCL5, as rantes derivatives do, and is a small chemical compound for which you could have development of resistance, which is normally a problem in therapy. And we could demonstrate that our peptides do still inhibit even this strain that is resistant to Maraviroc. And this is due to the fact that rantes interacts with its target receptor with a wider surface of interaction, and in this case the virus has more difficulties to adapt and mutate in order to enter the cells, even in presence of the inhibitor, because since rantes covers a large surface of the receptor, the virus basically cannot manage to adapt to overcome the presence of this inhibitor. And so this is a very big advantage in therapeutical for therapy and prevention. As a future perspective in the peptide derivatives of rantes, we wish to try a strategy in which we can multimerize these peptides, hoping to achieve an increase in avidity and therefore as a reflection in the anti-HIV activity. And we plan to do this in collaboration with Soledad Penades in Spain, and that is also a partner of the CHAARM consortium. And this is one of the main activities that we wish to do in the near future. Basically, there are advantages and disadvantages in the two rantes derivatives categories that are short peptide derivatives have in general a lower anti-HIV activity as compared to the full-length protein mutants, but they might have therapeutical advantages because they are smaller, so they might not be recognized by the immune system, and then maybe there could be no antibody response against these possible microbicides. Also, they should be more resistant to proteases because of their small molecular size, and they might have advantages for pharmacological applications, as the penetration in the tissues, also because of their molecular size, and may be more suitable for multimerization, again because of their size. On the other hand, the full-length mutants do have much higher anti-HIV activity, and so they certainly have a very strong interest. Both categories are very important in terms of being developed as CCR5 antagonists, which means that they should be able to bind to the receptor but possibly not activate it, in order to prevent pro-inflammatory conditions that would in the long term be deleterious both in therapy but also in prevention. And so these peptides are in fact antagonists, as Maraviroc is, and we are, as I said, we are working on a different context, also on full-length RANTES mutants, and we designed conceptual novel mutants. As I mentioned before, we are also working in a different context, still related with HIV microbicides, on full-length RANTES mutants. And in this context, we have already demonstrated the proof of principle for the possibility to engineer commensal bacteria in order to have them delivering these anti-HIV compounds in a strategy that is called live microbicides. And aside from the live microbicide approach, we also use this commensal bacteria as a platform to select new RANTES mutants, in which we could investigate new approaches to increase the anti-HIV activity, and we also coupled what is already known on RANTES mutants, taking advantage of the discoveries made by another CHAARM partner, that is Oliver Hartley, and so at the end we could fuse all the different mutations and couple all the different concepts, and at the end we could obtain a CCR5 antagonist that has an IC50 activity of around 5 picomolar. That is fivefold more potent than the best known RANTES antagonist at the moment. And in both field of research, we are also investigating several different combinations that are also within the aims of the CHAARM consortium. And we could see that we could have multi-target combinations as well as a single-target combination. And for single target, I mean that we could find that we could test at the same time different CCR5 targeted compounds with either additivity or even synergies. And this is very encouraging, because this opens also a new view on combinations. And even for the full-length mutants, we have several future perspectives to actually increase further the activity and try to enter the femtomolar scape, which would be, of course, a very important matter for developing extremely potent anti-HIV lead compounds.